Hello students, welcome back to the biology class. So far we in the last class learned about the electron transport system, or which is also called as electron transport chain, or the respiratory chain, or the electron transport mechanism in which electrons are transported with the help of complexes, which we learned in the last class. We learned that there are two shuttles which are possible for the transport of NADH2 into the mitochondria, and this whole concept involves either no use of ATP or one molecule of ATP is consumed. Accordingly, the shuttle number 1 does not consume any ATP, so we learned that that's why this will give rise to 3 ATP molecules, and the shuttle number 2, which involves FADH2, will require the consumption of 1 ATP molecule. That's why this mechanism produces just 2 molecules of ATP. We learned the constituents and the functioning of the ETS or ETC in the last class, that there are 9 electron carriers which are present in this concept, and out of these 9 electron carriers, 2 of them are mobile, and those mobile ones are coenzyme Q, also called as ubiquinone, as well as the other one is called as the cytochrome C. So these two are the mobile carriers we learned, and we learned that these electron carriers are grouped into 4 multi-protein complexes in the respiratory chain. The first complex is called as NADH coenzyme Q reductase enzyme, which transfers the electrons from NADH to coenzyme Q, and thus it reduces the coenzyme Q into ubiquinol. And we learned that the other complex is the complex number 2, which is called as succinate coenzyme Q reductase enzyme. It transfers the electrons from succinic acid to coenzyme Q, so it reduces the coenzyme Q into ubiquinol. The complex number 3, which is also called as reduced coenzyme Q, or you can even call it as ubiquinol cytochrome reductase enzyme. Its function is to transfer the electron from ubiquinol to cytochromes. Complex number 4, which is called as cytochrome oxidase enzyme, is responsible for transporting the electrons from cytochromes to oxygen. And we learned one main point, that the sequence possible will always have either the first complex or the second complex. Both cannot be present in the same pathway. So either the pathway of the electron is from first to third to fourth and finally to oxygen with the ATP formation with the help of complex 5. Otherwise, the next pathway is from second to third to fourth and finally to the oxygen, and then there will be ATP formation with the help of the 5th complex. So third, fourth, oxygen and 5th complex, these are the common things in both the pathways. The only difference is the beginning of the pathway. Either it will have first complex or it will have second complex. First complex play an important role when it is NADH2 involved, and second complex play an important role when FADH2, which comes from succinate dehydrogenase, that concept comes into picture. Complex number 5 is, we learned in the last class, is called as F0 F1 complex, or CF0 CF1 particle, also called as ATP synthase enzyme. We learned that there are 2 portions in this complex. One is F1 and the other one is F0. F1 is the ATPase enzyme, which is peripheral membrane protein, and F0 is the proton tunnel, which is the integral protein membrane protein. We learned that oxygen plays the role of the final acceptor of electron in the electron transport chain, and oxygen receives the electron from ETC as well as it receives the protons from the aqueous medium, and thus oxygen forms a molecule of water. So the end product in this, the role of the oxygen is at the end of the electron transport chain, or in the aerobic respiration. We learned that there are poisons also present which are possible, which can easily inhibit the respiratory process. We even did the concept of oxidative phosphorylation in the last class, that at each electron transfer, an electron donor is oxidized and the acceptor is reduced, and during this process energy is liberated, and that energy is used to produce ATP. It occurs inside the inner mitochondrial membrane of the eukaryotic system. It occurs in the mesosomes of the prokaryotes, and it was explained by chemi-osmotic hypothesis by Peter Michel in 1961. The only difference between the chloroplast proton gradient development and the mitochondrial proton gradient development is that in the case of the photosynthesis, we learned that the protons accumulated inside the lumen of thylakoid. Here the protons accumulate in the outer space. So in the case of the chloroplast, movement was from inside to outside, which causes the formation of ATP. In this concept, the movement is from outside to inside because there is more concentration of protons towards the outside, so it becomes acidic outside and basic inside. So protons move in this concept from outer space to the matrix, which liberates energy, which is used by, these protons move across with the help of F0 particle, and this liberates energy which is used by F1 particle to form ATP. We even did the calculation of total amount of ATP produced or kilocalories of energy produced from one glucose molecule. So we learned in the last class that one glucose molecule by aerobic respiration yields 686 kilocalories of energy, and we did the complete calculation keeping in mind that whether we go for the shuttle number 1 or shuttle number 2. In shuttle number 1, 38 ATPs are formed, and in the shuttle number 2 mechanism, 36 ATPs are formed.